Bismillahirrahmanirrahim. So we talked about the following so far. So we did an overview. So I gave you an overview basically of the 6 kingdoms, right? So we said the 2 bacterial kingdoms of prokaryotes, and these were the kingdom Archaebacteria and the kingdom Eubacteria. And then the rest of the kingdoms were eukaryotes, including the Protista, fungi, plantae, and Animalia, right? What we're going to do now is we're going to talk about each of these categories in detail. Now we're going to, in fact, talk about viruses and bacteria. Now, viruses, in fact, are not classified as living things by most biologists. But they're so important that we have to talk about them. And because they're the most simplest one in terms of their anatomy, it makes sense to talk about them first. Now, viruses being living things or not living things is controversial. But most biologists do not believe that viruses are living things. So example here is the measles virus. So viruses are much smaller than bacteria. Much smaller than bacteria. Examples here is the measles virus. See, it causes a rashy thing. You didn't get measles probably because you got immunized for measles, MMR vaccine, measles, mumps, and rubella. So we have vaccines for some of these common viruses. Okay, now you have to know what a virus is, okay? It's like, what is a virus? If I tell you what a bacteria, you have in your mind, Zai, you have something you can imagine, right? But what is a bacteria? What should you imagine when you hear a virus? You have to have a certain mental picture of a virus. Okay, easy peasy. A virus is basically some genetic material. Either it's DNA or RNA. And a protein coating to protect that DNA material. That's it. No, nothing else. There are DNA viruses and there are RNA viruses. In other words, all the virus will have is just DNA. And all the virus will have will be an RNA. So a virus will have some genetic material, DNA and RNA, and it'll have some protein coating. It's like packaged. You get, UPS mail effect package. Inside the packaged genetic material, that's a virus. Right? Now, if you just have a piece of DNA floating around, it's not a virus. What is it? It's just a piece of DNA floating around. Do you understand? Virus is simply some genetic material, DNA or RNA. There are RNA viruses. HIV. It's an RNA virus. Hepatitis C, RNA virus. Do you understand? So there are DNA virus and there are RNA viruses. So a virus is just a genetic material coming in a packaging of what? Protein. This packaging is called the protein coat. It's given a name. So here's 2 pieces of DNA, okay? You see that? Double helix. Cover it up with the protein coat like that, and you have a virus. Now, some viruses have the protein coating, and then on top of that, they have a membrane, like cell membrane. Do you understand? Some viruses have a cell membrane like this. So there are 2 types of viruses then. There are DNA viruses, RNA viruses. And then there are 2 types of viruses based on whether or not they have a membrane. Does that make sense? Therefore, if you were to cut up a virus, inside of it would be some genetic material, the nucleic acid with DNA, RNA, covered by protein capsid. That's the term you gotta know. The protein coat. So you say, what's a virus? It's like DNA or whatever. It has like no. Come on, we can make a sentence, a complete sentence, right? A virus has some genetic material, DNA, RNA, with the protective coating of protein called the capsid. This protein coating is called the capsid. And some viruses have an additional membrane envelope. It's called membrane envelope. Now, a virus can be as small as 4 genes. 4 genes. And they can have genes like hundreds of genes. Tiny viruses. Very big viruses. So that's the basic structure of a virus. Must know. They're even pretty cool geometric shapes, I might say, over here. Right? This is like a dodecahedron or something like that. Platonic solid. Side note, watch the videos on solid figures in geometry, because you'll see the beauty of this once you what I mean. This is a platonic solid. It's a perfect geometric figure. It's symmetrical from all sides. There are only 5 perfect geometrical figures like this in all of mathematics, and this is one of them. Now, so inside of it is a genetic material, and then followed by a capsid, followed by a membrane like this. Some have that, right? Now, on top of this, you see that stuff like glycoproteins. Glycoprotein means sugar proteins. Okay, you take proteins. Here's a protein. How's that protein? To protein. And you put some sugar on it. Sukar. You sugar on it. Protein plus sugar is a glycoprotein. Let's say, yeah? So protein that has some sugar elements attached to it, glycoprotein. Why is that important? Okay, watch. Okay. You got a cold, right? You got a cold virus, right? How come the cold virus doesn't go in your brain and cause meningitis? How come some viruses like hepatitis A causes hepatitis? It goes to the liver and like messes up the liver. How come it doesn't go to the brain or the kidneys, to the heart? How come? So how come hepatitis virus goes to the liver? How come the viruses that cause encephalitis, which is infection of the brain, goes to the brain? How does it know? And how come the cold virus affects the nasal passages and etc., but it doesn't go to the liver, etc.? Why? Okay, now let me tell you why. It's very important. Very important. All right? Pay attention. I was just telling the people before you here, whoever wants to go into the medical profession, whatever it is, you should listen to these lectures like however many times until you memorize this. Now, why does hepatitis virus goes to the liver and not to the brain? Let me ask you this. Does your body, do you think it's like a virus comes like knock knock, says, who's there? Hepatitis A. He goes, oh, welcome, come on in, take over my body and kill me. You think it's doing like, does it want to do that? No. Does the organism willingly invite the virus in? Yes or no? No. Yes. No, it's not going to invite it in, right? Okay. It fools the body to let it in. It fools the body. Do you understand? How? See, every cell has receptors, okay? The liver cell has proteins on top of it, like antennas, because it communicates with everybody else in the body. Every cell has proteins covering it receptors for other things. So virus is a con artist, right? It makes because glycoproteins that resemble whatever the other stuff that's supposed to attach to the receptors. Do you understand that? So by accident, hepatitis A virus discovered this protein that attaches to the liver cell. And the cold virus discovered the protein that goes to the nasal passages cells. It's sufficiently similar to whatever is attached to the receptor that it fools the cell to say, finally, I have mana. You see? Do you understand? I have what I was looking, right? So the glycoproteins are very important because they help infect the cells. So hepatitis A virus, hepatitis B virus, etc. like that, they infect their specific organs because they recognize, the receptors on those organs recognize the virus unwillingly. That's why viruses are organ specific. If ever the common cold virus gets smart, we're in trouble. Now, there are different types of capsids, okay? Capsids are protein covering. As I said, they're very geometric figures. There is the helical virus. In other words, that's the RNA inside of it, right? Outside of this helical structure is there to protect the RNA. That's its job. Do you understand that? They're called helical virus. Okay, like the tobacco mosaic virus. This is a virus that infects plants. Do viruses infect plants? Of course they do. They're like living things just like anything else. So, and then here's icosahedral. Icosahedral It's a platonic solid, one of the 5 perfect geometric figures. They have an icosahedral shape, okay? And then there's these viruses, complex viruses. They look pretty alien-type looking viruses. See that? You'll see, they get pretty, yeah? This is a bacteriophage. They got it down to a science, infecting bacteria, this virus. So there are different types of capsids. Now this is a tobacco mosaic virus. It's a virus that infects tobacco plants, which is of great interest to tobacco industry, which is a very important industry, right? Mucho mula, unfortunately. Terrible, terrible. As I said many times, there's some things in life you just don't want to mess with, okay? Because they've done nobody no good, right? That's one of them. And then this one example, this icosahedral variety, is the adenovirus. You know what the adenovirus is? Cold virus. Common cold. You are very intimately familiar with this one. Imagine has this beautiful geometric symmetry in a virus that causes such misery to you. And then there's the influenza virus, flu virus, right here. Cold. Now here's you know the difference between a common cold and a flu? Okay, common cold, a flu is like a truck runs over you. Oh, yeah, terrible. They're not the same. Flu is terrible. And this last one is called a bacteriophage. Phage is to eat. It eats bacteria. It infects. Now, this bacteriophage is important because the scientists use that bacteriophage to study the properties of viruses. So here's a bacteriophage. Now, why are viruses not living things? Why do so many people think bacteria are not living things? Because they need host cells to replicate. Because one of the 4 criteria of living things is they have to replicate. If a virus is sitting over there, it could be there for 1,000,000 years, it doesn't replicate. And people come, oh, that's a nice rock. It needs living things. If there were no living things around, there's no virus around. You understand? They are dependent on other things. Okay? So they need host cells to replicate. So that's why it's controversial, because they find living things, they replicate like crazy. As I said, viruses were studied, and bacteriophages were one of the viruses that helped in the study of viruses. So a bacteriophage, bacteria-eating virus that infects bacteria. Example here, T1, T7 virus, which infects the E. coli virus, Escherichia coli. Viruses need host cells to replicate. You must understand. So this bacteriophage is DNA over there, and then protein coding, right? Watch. So what it does is, see, it attaches like a spaceship, and then it drills a hole in the... Okay, to infect a bacteria ain't easy. Because why? It's got a cell wall. It has to drill its hole. Literally, it has to drill a hole in the cell wall, which is not easy. Okay, if you were a virus, it's not easy to drill a hole. But that's what this bacteriophage does, is figure it out. Okay? And the DNA just goes inside the bacteria. Like this. Watch. See, it's E. coli. See, it's attaching to the receptors. Are those receptors intended for the virus? No, they're not intended for the virus. But, the virus takes advantage of it. Does that make sense? Okay, so here's the virus. It's going to attach to them receptors, bacterial receptors. That's the bacterial outside membrane. Attaches it, see? And then releases the enzymes. Look, breaks open the digestive cell wall, and it injects its DNA into the bacteria. Now, that's kind of freaky, right? It's kind of like alienish, right? I think so. But that's what it does. Okay, replay. Watch. So, it attaches to the outer membrane. Some bacteria have 2 membranes, like this over here. On the receptors, which are not intended for the virus, right? Of course. You understand? And then it drills a hole in the cell wall. This is a cartoon picture. Cell wall can be pretty thick. And then it injects the genetic material. Does that make sense? Right? Now, where do those enzymes come from? Inside. They're inside the capsid. Yeah. Some bacteria have some extra enzymes. You follow? Okay. Some viruses. Okay. So, viral attachment to host cell to the cells they're going to infect, is a specific process. Different viruses recognize different attachment proteins. Does that make sense? Now look at this. Now this virus, look at the, I just want to make sure you understand. This virus here does not have a membrane protein on it, right? No, a membrane around it, right? This virus does not have a membrane envelope. Do you understand? It does not have a membrane envelope. So that's how it gets in. Now this virus has a membrane envelope. Look how it gets in. Just watch. That membrane envelope has antennas that recognize the receptors. You see? So the ones that have these membranes, their own membrane becomes part of the membrane of the whatever the host cell is. Does that make sense? Those that don't have the membrane, they have to find a way of digging in there, right? You follow? Now, what do they do when they get inside? Well, they take over is what they do. Look, now here's a virus that's inside now. Since membrane's gone. It doesn't need the capsid. Capsid gone. Okay? So once it gets inside, what does it do? It takes over the cell. It's like, I'm the boss. So I'm the boss. I'm going to kill. No way. It takes over the cell. What do you mean takes over the cell? Okay, to make new viruses, what's it going to do? It's going to make like gazillions of new viruses, okay? Do you understand that? To make new viruses, you need to have more of, make copies of the DNA, right? Or RNA, right? Right? Now, as I said, the virus is a simple structure, right? Now, to make DNA or RNA, you need ribosomes, you need nucleic acids, you need all of that stuff, right? Where is it going to get it from? Everywhere in the cell. It's like colonizing a wire, you know. Say it's all the natural resources now belong to the virus. Do you understand that? Everything it needs to make more viruses. Okay, the DNA and RNA that's going to make new virus DNA RNA would have been used to make more cell DNA RNA. Do you understand that? The DNA RNA that are made for the new viruses, the nucleic acids that are needed to do that would have been used otherwise for the cell's own purposes. And the machinery to make new DNA RNA is the cell's machinery. And the envelope is the cell's envelope. Everything is like eating up the cell from the inside out. Does that make sense? So the first is it has to get inside the cell, right? How's it going to take over from the inside? It's like you're standing outside the castle. Now I've taken over the castle. You're on the outside. You gotta get inside the castle. Do you understand? So first has to get in. Once it gets in, the capsid dissolves. Okay? Or in the case of the bacteriophage, for example, only the genetic material enters. Do you get that? Okay. Now once it gets inside, it does one of two things. Either it goes in there, takes over, makes a boatload of viruses. And all these boatload of viruses exit the cell and they kill the cell. Or it gets inside, goes into stealth mode. What kind of mode? Stealth mode. Underground, subterranean, secret, secret society. The second thing that they can do is stealth mode. There you go, stealth mode. What they do is the genetic material goes in there and becomes part of the host genome. See? Here's the stealth mode. Genetic material becomes part of the host genetic material and then stays there. And every time the bacteria replicates, for example, the virus genetic material replicates. Does that make sense? So it stays there for a long time, sometimes years. So the first thing it can do is take over, make a bunch of baby viruses, and boom! That's called the lytic. Lytic is to break. So lytic cycle. Lysogenic cycle is what should remain dormant. So the first, if it kills the bacteria by making a whole bunch of baby viruses, or kills the cell, whatever cell it invades, that's called the lytic cycle. If it stays dormant, that's called the lysogenic cycle. Does that make sense? Watch. This is the lytic cycle. It's going to take over, make baby viruses, and the thing is going to explode. The DNA, genetic material, penetrates into the host. This is a bacteria because they have circular DNA. DNA is copied using the bacteria's machinery. Capsids are synthesized using bacteria's machinery. Bunch of viruses are made and they break out. This is the lytic cycle. Now, lysogenic cycle, the dormant cycle, watch. Comes in, virus comes in, is the viral genome is going to become part of the genetic material of the host. In this case, this bacteria. It's going to go in there. This freaky alien-looking type of virus. It's going to take over. Not yet. It's going to become part of the gene. Now, every time this virus replicates, it's going to replicate the virus's genome. And then years later, sometimes, or a long time later, whenever, it can do what? It can come out with a vengeance. Replicate. The viruses then don't enter cell because they want to go in the lysogenic cycle necessarily, right? They just want to chill it. They come out and they break eventually. Does that make sense? Yes. That's a very good question. Oh, that's a very good question, actually. Okay, I want to make a bunch of handouts, okay? I want to make a bunch of handouts. So I give you a sheet of paper, okay? 2 sheets of paper. I want you to make, okay, 50,000 copies of this. It's going to take you a while, right? But then I say, okay, I want you to make 5,000. I want you to make 5,000. I want you to make 5,000. You make 5,000, 5,000. Who's going to be faster? The second group is going to come back faster, right? So one virus becomes part of the bacterial DNA. The bacteria divides. Now you have 2 copies of the virus genome. Bacteria divides again. Some bacteria divide every 20 minutes. So come back a year later, come back 1 hour later, you have like 6 copies of the viral genome. Do you understand? You follow? I'm guessing that's speculation. Of course, we can't really ask a virus because they don't have sensory receptors. But that's a theory, I guess, speculation. Does that make sense? It's much easier because all they want to do is replicate. How does it? I mean, it's fascinating. Some of these questions are more complex, metaphysical type questions. Why do they even do that? Why do they have to do it? Why do anything have it? Why do we have to have plants? Well, you know what I'm saying? They do it because that's part of the Lord's creation. Allah made it that way. Yeah. Okay. How do they exit? They exit like this. Watch. They go out there like bubble burst and they exit. Now this virus, like this, it captures the membrane envelope like this. This virus already has a membrane. It's just going to exit, okay? This virus is going to capture the membrane of the host cell and then exit. Now, we fight back viruses, right? Because we have immune cells that recognize, hey, you can't fool me. So immune cells fight back. So immune cells have receptors for viruses. And they take in the virus. And then they attach it to the lysosome and which destroys the virus. That's how immune cells work. Immune cells have the receptors that the viruses have. There's viruses and there's retroviruses. Retrovirus. The most famous retrovirus? HIV. Now, retroviruses are very peculiar and interesting. Now, look. Retroviruses are this. They are RNA virus. What they do is they come in as RNA, but then they change into their DNA complement. So they come in as RNA, they make DNA out of RNA. That never happens ever otherwise. It's always DNA makes a copy, and that's called RNA. RNA goes to make proteins, right? This never happens otherwise except in retroviruses. You don't make DNA out of RNA. RNA has no business making DNA. Do you understand? It's like, astaghfirullah. You can't do that in bio. Do you follow? RNA has no business making DNA except in retroviruses. Do you understand? Retroviruses, like HIV virus, they start out as RNA and they make a DNA out of it. They go backwards. And then that DNA becomes part of the cell. Because RNA is not going to become part of DNA, right? Now, otherwise, if a virus is a DNA virus, it enters the cells, can go straight in the nucleus and become part of the host DNA, right? But what do you do with the virus that's an RNA virus? It says, can I come in? No, you can't come in. So this is the nucleus. You're RNA. Do you understand? But it comes in RNA, makes a DNA copy, and the DNA enters the nucleus and becomes part of the host genome. Does that make sense? This is HIV. This is what HIV does. Okay? So HIV RNA becomes DNA, that becomes part. Now, and then when it's time for it to cause trouble, that's AIDS. See, HIV infection, in the initial stage, it's called HIV infection. When it's destroying those cells. See, the HIV virus affects not liver cells, doesn't affect kidney cells, doesn't affect this cell. It affects immune cells, the very cells that are supposed to destroy them. That's why it's particularly dangerous, because the immune system is like shut down, right? So when HIV virus becomes lysogenic, it comes out of hiding, it causes all the immune cells to die, then the person becomes susceptible to all kinds of infection. That's AIDS. There's specific criteria for AIDS, correct? So somebody has HIV infection, okay? Somebody could have you wouldn't be able to tell. Somebody's got AIDS, they're probably in the hospital because they're fighting some kind of infection. Do you understand what I'm saying? So that's retroviruses. Does that make sense? Retro means going backwards. Watch this. RNA giving DNA. That's like weird. RNA. This is a cartoon picture, of course. Making DNA. Same thing. RNA becomes DNA. Okay? And DNA becomes part of the host DNA, and when it wants to, it becomes active and makes this. This is HIV virus. Okay? Now watch. HIV virus. This is actually HIV virus. Infects the cell. Now look at it. It's kind of rolling like a ball, right? And all those receptors, it's like Velcro. All those receptors slow it down and eventually attaches and it stops. Do you understand this? And then virus, you see what's happening? It loses the envelope, right? You follow? And then inside is the capsid and the virus. It melts into the cell. Everybody understand this? It melts into the cell. And once it melts in, the capsid dissolves, right? And then it goes and becomes part of the material of the cell. And then when it reactivates, there it goes. Does that make sense? This is shingles. This is what I was taking. This is a lysogenic cycle. This kid is the same kid over here in the other picture. This old man is the kid, 60 years later. Because this virus went dormant in him for that long time. Initially, he had chicken pox. Chicken pox, he got better from it, but some of the virus went dormant. It likes to hide in nerves. That's why it affects in a peculiar fashion. When it comes back in the distribution of the nerve. So this is shingles. Virus is also bad, like this one. This is a hepatitis B virus, and this is one of the most commonest cancers in some part of the world, like in Egypt, for example. It's very, very common because they have a lot of hepatitis B. And it takes over the cell and anything that messes with the genetic material is not good, right? Yeah, it's high risk for cancer. So this is liver cancer in somebody who has this virus, see? That's normal liver tissue around, and that's liver cancer. So viruses can cause cancer. On the other hand, now this is a tobacco plant. That's what they make cigarettes out of. And this virus infects. Obviously, these leaves are not supposed to look like that. Nobody's going to buy cigarettes that look like that. I hope all of them. I really hope actually that all the cigarette industry plants like this get destroyed someday, because it's really. And why would you make something that's poison for other people? I've never seen a patient with pancreatic cancer except one time. I've seen a lot of pancreatic cancer who was not a smoker. And then tulips. You've seen tulips like this. They're colorful like that because of virus. A virus infection causes them to have colors of this sort. So they can actually, they're called Rembrandt tulips. Rembrandt was an impressionist painter. Beautiful, colorful tulips. So not all tulips, not all viruses are bad. Until next time, as-salatu was-salamu ala rasulillah wa alhamdulillahi rabbil alameen. Assalamu alaikum.